Okay, pick up your notebooks first.
Are you ready? Yes.
Not test, but you have to write down question answers. So get ready with your notebooks. Oh, it's not opening.
What
is that? It's not happening. Write one time. Write one. Okay, first, what are we going to study today? I'm going to give you questions. क्या हो गया? तुम्हारा हाथ पकड़ रहा है? इसका हाथ मत पकड़ो, उसका हाथ पकड़ो, तुमको कुछ नहीं मिलने वाला. Now, we are going to study some major terms. First of all, if you remember, we had already studied what the other day. We stopped with what is the condition of a turgid thread and names like turbo pressure, wall pressure, etc. So we start from there. First of all, we talk about what would be the condition like. We talk about turgid, and then we talk about turgid, and then flaccid, and then plasmolite. Let us try understanding this first. No, it's not happening. It's not reading. This one. Huh? Where is it? Here. This one. This one. Which one? It should open from here, no? Arey baap re. कौन जा रहा है? हाँ? What are you doing there? The first one, fatafat, we'll start answering them. The first one is, why are root hairs unicellular? What should you write? Why are root hairs unicellular? Why are root hairs unicellular? Bolo. No, why are root hairs unicellular? Unicellular. because it is just an extension. The entire root hair cell, it may look quite big, but it is a single cell, unicellular, because it is the extension. How do you draw the root hair? The root hair cell, a single epidermal cell का ही extension रहता है. Never mind, it's quite a big structure, long structure, but it is a single big cell. Complete करो फटाफट. Why are root hair cells unicellular? Because it is an extension of a single epidermal cell. This should be cell, epidermal cell of roots, epidermal cell of roots. The two outer layers of root hairs. What are the two outer layers of the root hair cells? Two outer layers of the root hair cells are what? Two outer layers of any cell wall, cell membrane, like any other plant cell. Cell wall, cell membrane. This you know already. This is just a summary of what we have done the other day.
Movement of molecule from higher to lower concentration in that cell.
My main question is the absorption
The movement
of
of
the
the
water by living
One
organism.
at a time.
Absorption of water by living organism and plants or animals.
By by surface attraction. By surface में higher concentration, lower concentration नहीं लिखना है. Semi-permeable membrane नहीं चाहिए. So this is what you have to remember. Phenomena by which living or dead cells absorb water by surface attraction. By surface attraction. Examples, many we had discussed. You should remember those examples. Definition with examples are often asked in your exam. Be serious, Kanna. Next, osmosis should be defined as. Now this you already know. Durvesh bolo.
Yes, now the movement of molecule from higher to lower.
Movement of molecules of
Water
हाँ,
from higher to lower concentration through a semi-permeable.
very good. यही लिखना है. Done. Osmosis का definition खुद लिखने की कोशिश करो. See that you are able to write on your own. Now, next is done. No, all of you done?
No,
Make it fast. Over. So, the first, the second, the third. Try to understand what you are writing. Definition of osmosis, you must have got it by heart so far. Sure. Now we move on to the next. Endoosmosis means endoosmosis means Hypotonic solution में क्या हो रहा है? What is happening to that cell? It's swollen up. So where is the water going? From where to where? Water is entering the cell or coming out of the cell? Entering the cell. When the cell is placed in a hypotonic solution, when a cell is placed in a hypotonic solution, the inward movement of the water in a cell, the movement of water in a cell, inward movement, that is called endoosmosis. Inward movement is called endoosmosis. Done. Semi-permeable membrane, 2 examples. Lifting back, muslin cloth में, lifting back, सबसे important दो, cellophane और animal bladder, lifting back, parchment paper, cellophane membrane, any of the two you write.
Now about that egg also.
Egg membranes. Egg membranes. Come to the next one, number 7. Read question number 7. Sugar cubes are dropped in a beaker containing water. The water slowly becomes sweet. Which phenomena?
Diffusion.
Diffusion.
But non-permeable.
Impermeable. Non-permeable में होता impermeable. Impermeable. You may write till here. This you need not write. This is very simple. Just to understand. What is semi-permeable membrane now? The definition of this is very important.
Membrane through which solvent can pass around.
Correct. Substances, selective substances will move. Only the movement of molecules of solvent will be allowed. No movement of the solute. It prevents passage of solute molecules. ये लिखना है. All of you should write this. You know the definition of semi-permeable membrane. You should know the correct definition of this. Who is this? Obviously, Shandy, no? Shandy के अलावा तो
Please describe it.
done.
Seven.
Chalo. Now, okay, this you will write after some time. Now, next, what are we going to study is we are going to understand the conditions of turgid flaccid. Let us try to understand from here again. We were discussing this the other day. So let us start from here. So here, what I'm trying to show you here is the conditions of turgid and flaccid. The last class already we were talking about. If I talk about a cell, if I'm talking about a cell, the cell which is absorbing water, the vacuole is filled with water, the vacuole is distended with water. Under this condition, if you see that the vacuole is quite big enough, the cell wall and the cell membrane are almost pressed towards each other. Is there a wide gap between them? No, they're almost pressed, they're almost very close, but they generally don't get fused, but they are very close, they are pressed against each other. And then this entire vacuole, which is filled with the sap, is making the cell also quite tight. How do we understand? Yes, by looking into the plant parts, we can understand that. By looking into the leaf, by looking into a flower, we can always understand whether the cells are turgid or not. The other day, I was also telling you with reference to aquatic plants also. So let us now just understand two things, that I am talking about a cell which is turgid. What is the meaning of the word turgid? A cell, the state of a cell, which has already absorbed. Sit straight. What's your name? Sriram, sit straight. You have not come to his house that you are chatting and face to face, you are sitting and chatting with him. No. So now that I talk about this particular cell, a cell is turgid. What is the meaning of the word turgid? Condition of a cell, the state of a cell which has already absorbed a lot of water, a state of a cell where the vacuole is distended. What is the meaning of distended? It is so wide, it has already taken in so much water, now it is no more in a state to absorb any more water. Endosmosis almost band ho chuka hai. Q, the vacuole has reached its last limit to absorb water. But are we to understand कि इस condition में the cell will burst? No, the cell will not burst. अब जब the cell is already distended, अब जब the vacuole is filled with water, there is enough amount of pressure exerting from here towards the membranes. From here towards the membranes. मतलब center से periphery तक pressure is getting exerted. Center से periphery. Why? Because the contents of the cell has become so much. The vacuole is filled with water. But again, I repeat, under this condition, is the cell bursting? No, because the wall, that is the cell wall and the cell membrane, will also start exerting pressure into the center of the cell. They will also start exerting pressure on the center of the cell. So there are two pressures counteracting. एक जो है center से wall तक जा रहा है, which is known as turgor pressure. And there is a counteracting force from the wall to the center, जिसको बोलेंगे wall pressure. So these are the two prominent, there are some others also, but these are the two prominent pressures that you have to understand right here in this situation. Why does a turgid cell easily don't get burst? Are we to understand the moment it is turgid, it's bursting out? No, never. It will not burst so easily because there is always a counteracting wall pressure. So एक है center से wall तक turgor pressure, wall से center तक wall pressure, and both of them are counteracting each other. This prevents the bursting of a cell. This prevents a cell from getting destroyed, from getting burst out. So if this is what it is, now we are trying to understand under what type of a condition will you get up from there. Now see, in this classes, एक तो एक ही घंटे का lecture होता है. You are not sincere with your work. That is your problem. आई एम वेरी सिंसियर विथ माई वर्क, आई एम ऑलरेडी डिफाइंड कि इस लेक्चर में मुझे यहाँ तक खत्म करना है. बट इफ 10-15 मिनट्स तुम लोगों को डिसिप्लिन करने में जाएगा, देन इट्स वेरी डिफिकल्ट टू फिनिश अ कोर्स. इट्स वेरी डिफिकल्ट. You are in the classroom. You are in the classroom. Why the teacher has to tell you to study? Why your parents have sent you such a big body? You understand? No? If at all you have brains. Why should I tell you that you concentrate, you study? एक और बार I see a disturbance. Shri Ram and both. I already warned you. Sit straight. I warned you. I don't give too many chances to people. एक बार बोल दिया खत्म है, second time दोनों बाहर हो जाए खत्म. I don't want any disturbance in my lecture. Simple as that. Now here what I said. This is a condition. How will a cell be turgid? If you can tell me, under what condition will a cell be turgid? Under what condition a cell will be turgid? What should be there in the surrounding medium of the cell? Water. A hypertonic medium should be there. सेल के साइटोप्लाज्म या वेक्यूल से ज्यादा अगर डाइल्यूट मीडियम सराउंडिंग में होगा, then only the process known as endoosmosis will happen. Then only the process known as endoosmosis. Just now we had answered a question. Endoosmosis मतलब inward movement of water. Endoosmosis मतलब inward movement हो रहा है. Now that there is so much of inward movement, that's the reason the cell is becoming turgid. That's the reason the cell is becoming turgid. Now what do? We pick this cell and put in a sugar solution. We pick this cell and which is a turgid cell already. We pick this turgid cell and put it in a concentrated medium. So ये concentrated medium को क्या बोलेंगे? Hypertonic medium. This is a hypertonic medium. In a hypertonic medium, what will be the behavior of this cell? Endoosmosis दिखाएगा कि exosmosis दिखाएगा? Exosmosis. This cell will show the process of exosmosis. What is the definition of exosmosis? Endoosmosis. The reverse of it is exosmosis. Outward diffusion, outward movement. So इसमें क्या हो रहा है? In this cell, the process of exosmosis is happening. But if the process of exosmosis happens for a long time, then what are we going to observe? Will the cell remain turgid? No. The turgidity will get lost gradually. The turgidity will get lost gradually. पहले जो vacuole was fully distended, was quite big enough. Now the vacuole has started becoming shrunken. Now the vacuole has started becoming shrunken. Why? What has happened? Exosmosis has happened. This condition is known as flaccid. Flaccid, remember we are talking about the state of a cell. And if I have to talk about flaccidity, flaccidity is the condition of a cell. So how do we define the word flaccid and flaccidity? What is flaccid? The state of a cell which has given out a large amount of water by the process of exosmosis. The state of a cell which has given out a large amount of water by the process of exosmosis, and the condition is known as flaccidity. The condition is called flaccidity. The cell, how is it? Is it taking in water? Then it is turgid. Is it giving away water? Then it is flaccid. State of a cell. So now that we are saying that yes, if the cell is in a hypertonic medium for a continuous period of time, then the cell is showing exosmosis and the state now becomes flaccid. But if this continues for a long period, then what? The cell is still in the hypertonic medium for a long period. The cell is still in this hypertonic medium. I'm still saying the cell is still placed in the sugar solution for another 3-4 hours or even a longer period. What are we going to observe? In this cell, we are going to observe that the process of giving out of water by the process of exosmosis will keep on continuing. The process of giving out of water by exosmosis will continue. And if it continues, then what are some of the changes that we are going to come across? The changes that we are going to come across would be the vacuole would be extremely shrunken as compared to what you know as flaccid. Flaccid way, the vacuole still has got an adequate amount of sap in it. But the moment that we talk about a condition where the cell has given out so much of water by the process of exosmosis, then you are understanding that exosmosis is a condition which is constantly going on and the cell is still placed in a hypertonic solution, the vacuole will become shrunken. What is the name that we understand? The cell membrane and the cell wall. The cell membrane and cell wall are separated, are wide apart now. They are getting wide apart. And what will also happen to the cytoplasm? The cytoplasmic volume will reduce. The cytoplasmic volume is extremely reduced. So when all these things are happening, the cell is becoming smaller, extremely shrunken. But actually the outer shape of the cell does not easily get disturbed. It is actually the cell membrane, the cytoplasm and the vacuole. The cell wall is made up of cellulose and it is quite tough. The cellulosic layer that we understand is made up of sugar and that is quite tough a layer. So the cell wall is the last thing to get disturbed. So cell membrane will get disturbed. Cell membrane and cell wall बहुत ज़्यादा distant apart हो जाता है. What about the cytoplasmic content? The cytoplasmic content also will come down, shrunken. What about the vacuole? The vacuole will thin down. If that is what it is, the state of the cell, then this state of the cell is called plasmolized. So plasmolized state of a cell is an extended condition of or an extended state of flaccid. Flaccid condition also we have seen that the process of exosmosis happen, and here also the process of exosmosis is happening, but for a longer time. When a flaccid cell undergoes the process of exosmosis for an even longer time, then only the cell undergoes plasmolysis. So what is plasmolysis? A condition when the cell is almost on the verge of dying. Plasmolysis, the cell is almost on the verge of dying. The cell is almost on the verge of dying. Why? Because the cytoplasm is extremely shrunken, dried out almost. The vacuole is extremely small, becoming extremely dried out, reduced, shrunken. The cell membrane and cell wall wide apart. And what is filled up between the cell wall and the cell membrane? The cell wall and the cell membrane is now filled with the hypertonic solution. Why? Because the cytoplasm has lost so much of water. It is only some proteins and ions and sugar left in it. So that's the reason we always understand in a plasmolized cell, water content बहुत कम हो चुका है. But if you want to save this cell from dying up, immediately what can we do? Immediately place this cell in the water. In a dilute solution, place this cell, which is almost in the process of dying. That is what we do, no? If I talk about the plant parts, which is almost dying, what do? We water the soil. And then we may see that yes, the leaves have again become fresh. Sometimes they may not become fresh because quite a number of times if the cells have already died out, do we get to see that the plants will again revive? Sometimes they may not revive also. When the cells of the plant parts have already died out, then they will not revive. But if they have revived, that means they were not fully dead. Then I say that how can we revive this plasmolized condition? The plasmolized condition, cells are to be put in water or a dilute medium. When the cells are to be put in water or a dilute medium, the plasmolized cells are put in a dilute medium, then what do we get? We get the cells reviving again, and this is what is known as a deplasmolized state. What is deplasmolized or what is deplasmolysis? The reversal of plasmolysis. So now that you put the plasmolized cell in the water, what will happen to this cell? This cell gradually will try reviving. How? This cell gradually will now try taking in water. The cell wall and the cell membrane will now again start coming closer, and the vacuole will again start becoming bigger. The cell has not died out, and we could be able or we are able now to revive the cell. What did we learn? We are talking about two terms, plasmolysis and deplasmolysis. What is the difference? Plasmolysis is the extreme loss of water by a cell placed in a hyperchronic medium, and the extreme loss of water is by which process? Exosmosis. Bolo.
Ma'am, if the plasma cell is dead and ma'am we put in a hypotonic solution, ma'am still the water, the endosmosis will occur and the cell will become turgid, but it's dead.
No, that's what I said.
Inhibition
When we are no inhibition cannot revive the cell fully.
but it will take place.
It will take place, but it will not be able to successfully make the cell turgid again. But when we are talking about, just now I said also that I'm talking about the small plants that we have in our house, and quite often we get to see that the leaves are drying out, and we may be putting sufficient water in the soil at that time. But sometimes we get to see that even then the plant parts or the leaves do not revive. Even then they must not be reviving next morning. If that is the case, then what are you to conclude? By the process of plasmolysis, the cells have already died out. But quite often, when I get to see that the leaves in my window sill, they're getting dried out, they're getting rolled. What I do, I put some water. And next morning, I get to say that they are again revived. And what is the conclusion? The conclusion is, yes, the cells were undergoing flaccidity, the cells were undergoing plasmolysis, but they were not fully dead. So can we revive a plasmolized cell? Yes, by putting the plant parts in a dilute medium. But if they are fully dead, plasmolized means not necessarily plasmolized cell is a dead cell. On the verge of death, yes. So if before they are completely dead meaning what? The water content of the cell should be finished. So before that is done, if before that is happening, if you are able to successfully put the plant parts back into water or in a dilute medium, we can revive that condition. So that is what is called deplasmolysis. You get questions, how can you revive a plasmolized cell? A plasmolized cell can be revived by putting the cell in a hypotonic medium, by putting the cell in water. Bolo.
But basically plants, so ma'am, plasmolizing takes place due to transpiration.
No.
But they don't die by putting a hypertonic solution.
When we are talking about plasmolized condition, plasmolysis of the plant parts may happen not only because of transpiration.
In a potted.
Even in a potted plant, say suppose even in a potted plant, the soil water is inadequate and the weather conditions are hot and dry, then also the cells may die out.
That due to transpiration.
That is due to transpiration, but transpiration along with dry condition of the soil. Transpiration along with wet condition of the soil, balanced it is balanced. But yes, if the environmental condition is hot and dry, of course transpiration is happening. But I may also understand that the potted plant that I have, what am I doing is I'm adding some manure. But when I've added so much of manure or some fertilizer, the entire soil medium is becoming extremely concentrated, but I have not added too much of water there. Then also the plants may start dying out. So when I talk about what may be some of the reasons for plasmolysis, say suppose I talk about a farmland, a garden, a lawn, grasses are there. I take a handful of salt and sprinkle over the plants. What will happen? Are these plants going to flourish or they are going to die? They are going to die. Why? The soil water is becoming hyperchronic at the time because I have sprinkled salt. And since the soil water has become hypertonic, the plant cells, the root cells, instead of taking water, they have started giving out water. They have started giving out water, even though the environmental conditions may be moderately humid, even if I say that. Moderately humid condition may be the plant cells may undergo plasmolysis. What are the other conditions? By applying fertilizers. By applying fertilizers in the land, in the farmland, apply fertilizers, excessive fertilizer, and the amount of water much lesser, irrigation less. Is that a reason for the plasmolysis of the crops? Yes, that also. What is the other characteristics of plasmolysis? Are we benefited by this process of plasmolysis? Kaise?
When there are algae.
When?
And when algae is growing rainy season, we apply chlorine on it.
We apply chlorine on it. So yes, the cells, the single cell organisms are dying out of plasmolysis. We have a throat pain, we gargle with saline water. What are we trying to do there? The area inside our body, inside our throat, which may be having some bacterial infection. The moment you are gargling always with saline water, what's happening? You, after gargling for 4-5 times, you get relief. Q, because whatever amount of bacteria was there, that's not able to multiply, because every time you are gargling with the saline water, the surrounding medium of the bacteria becoming hypertonic, and they are releasing water. They are giving out water, the cells are dying out by the process of plasmolysis. So that's the reason whenever we suffer from any type of an infection, cuts, wounds, there's always a habit. Wash with saline water. Why? Because whatever germs, pathogens, microbes are there, they will be died out because of the process of phenomena of plasmolysis. Now we again come back to this word, turgid. What is the meaning of turgid? And we try to see some uses of turgid condition of the plant. What are some of the benefits of the turgid condition of the plants? The benefits of turgid conditions of the plants. The first one is we talk about the stiffness of the plant parts. The benefits of turgid condition we are talking about. The first is the stiffness of plant parts. Now this quite a number of times I have been bringing in with reference to the process of turgidity, turgid condition of the plant, turgidity, condition of the leaf, turgidity. Now, when we are talking about turgidity, I always mention that are we able to understand the turgid condition of a plant, turgid condition of a flower, turgid condition of the leaf? Yes, we are by looking into the plant. If the leaves are spread out, they look fresh, they look shiny, quite a number of types of plants. If I talk about the money plant, yes, the leaves look shiny and spread out. And when the water is less, how does the leaves look like? Rolled. They yellow also. If they have already started yellowing, meaning they are almost dead, can you revive them by adding water? No. Abhi deplasmolysis nahi ho sakta hai. So that's a very clear indication. The types of parts of the plant, leaves especially, whether they are turgid or they are flaccid. Whether they are turgid or they are flaccid. So this is what we are trying to understand. Stiffness of the plant parts. Now it is not only for the leaves, it is also for the young green stem. For young green stem also the same basic condition would apply. But should it apply for bigger stems, thicker stems? Should the condition of turgidity should apply for bigger stems like the mango tree, the coconut, the betel nut, the palm that we get to see? No, because they are already big, broad trunks there. The outer layer is already made up of sclerenchyma, dead tissues there. So the process of evaporation much less. So the cells already are built up in such a way that they can conserve water. Okay. So if I'm saying which are some of the plant parts where you can easily understand whether the plant parts are in turgid or flaccid, never to write roots. Young green stem, ya to leaves, flowers, the flower petal, the petioles and the stalk. Water come over, they start drooping, and water अच्छी तरह से रहेगा, adequate amount of water will be there, then they would be stiff. Say suppose when I talk about the petiole, the stalk of the flower also, the stalk of the flower, the petals of the flower. So that is what we are talking about, stiffness of the plant parts. Let us talk about another one. Germination. Let us talk about this, germination. Now, when we are talking about germination, what happens? When I talk about germination, what happens? The seed is under the soil. And when the seed is under the soil, are you able to see that side? The seed which is under the soil, what would happen to the seed? The seed is under the soil. The seed coat which is dead is absorbing water by? The seed coat which is dead is absorbing water by imbibition pehle. The seed has not yet developed root hairs. The seed has not yet developed roots. So pehle to the seed has got seed coat which is the dead part and it is absorbing water only by imbibition. Now the cells of the seed, they are becoming turgid or flaccid? Turgid. So seed coat absorb kar raha hai water. The cells are becoming turgid and iske reason se, iske wajah se Now some additional tissues have started developing because the process of cell division now can happen. But if adequate amount of imbibition does not happen, if the soil is dry, and the seed is in the soil, will the process of imbibition happen? No. So this seed which is in the soil, first of all, there should be adequate amount of water in the soil. There should be a adequate amount of moist environment around the seed coat. Now it is absorbing water. Cells are turgid. Additional tissues develop in the form of roots, fine roots. So अभी जब ये roots निकला है, they are going deeper into the soil and now they are absorbing water. Now the cells of the seed are even more filled with water. A turgid condition is happening now. The cells of the seed are even more turgid क्योंकि एक तो seed coat से imbibition हो रहा है and root hair से कौन सा process हो रहा है? Root hair से osmosis हो रहा है. So now for both the processes, the seed cells are fully turgid. Now that the seed cells are fully turgid, now the seed coat starts bursting out. The seed coat has already started bursting out a little bit and now the seed coat starts completely getting bursted out. And ये जो छोटा सा एक plumule develop होता है, the plumule which starts developing. Now that I talk about the seed, if this is the seed, first of all we talk about that the plumule and the radicle. How does it develop? The plumule and the radicle. So first of all, the fine root hairs. But this part of the plant, it will start pushing the ground. This part of the plant will start moving this way because this is known as the radicle. And this is the green part which is known as the plumule. And this will start pushing the ground and the growth has started from here and it will start moving up. The plumule will always start moving up. But how is it moving up? Because these cells here are so very turgid that they have that much of pressure to move against the gravitational force and push the soil and start flourishing upwards. So धीरे धीरे क्या हो जाएगा? They will start breaking through and they will start moving up like this. They will start moving up like this. So ये जो process हुआ है, that is pushing through the hard ground and the growth of the plant happening. This is only because of the extreme turgid condition of the cells that the pressure has developed there. Did you ever come across that the big roots of certain plants on the roads, they break through the wall and go, they break through the wall. Quite a number of times we come across that the big trees which may be on the other side of the wall, maybe somebody's courtyard, somebody's house, and they have started breaking the wall and running over the footpath. So quite often we have seen this. What type of condition we have to understand? Turgidity. The turgid condition is responsible for this. So seed germination is one characteristic that we always understand. Next is we talk about root pressure. This is very important. Try to understand what I'm saying. Root pressure. What is root pressure? We learn the definition. First of all, let us try to understand one activity here. Now here, to understand the concept of root pressure, we are saying that if this is a particular plant, then it has got the roots, the tap roots. It has got the tap roots and all the root hairs and the large number of the root hairs, etc. Now that we are talking about the stem is constantly absorbing water. The roots are absorbing water. So what is the directional movement of the water? By the way of the roots, and the water is moving up in this direction. By the way of the roots and the water is moving up in that direction. How do we understand that the water is moving up? What is the proof that the water is moving upwards? Yes, we are theoretically very much understanding that yes, in the leaves, the water reaches and the process of photosynthesis is happening. But if we have to find out experimentally, does water actually really move up? Yes, especially for the smaller plants. If you break the stem, did you ever come across some clear liquid oozing out from the stem parts? Yes, we have always seen. So take a small green plant, a fresh plant, young green plant, and break from the stem region. And in just a couple of seconds, it will come some clear liquid is oozing out. That process is actually called the bleeding of the plant parts. Bleeding because you had caused an injury to the plant part. No? Injury and oozing out body fluid for us is also bleeding. So same for the plant also, the word bleeding is applicable when you are breaking the stem, young green stem, and the clear fluid coming out, that is also called bleeding. But experimentally, if that has to be done, then what do we do? Maybe we take a young green plant, and in this young green plant, we cut out the stem portion this way. Keeping a definite length, cut out the stem portion. And over the stem portion, we are to fix this glass tube. We are to fix this glass tube. Fasten it with a rubber tubing here. So with the rubber tubing fastened, and then this glass tube should be in a position. Then what? So if I'm saying that if this is the cutout portion of the stem, then what are we supposed to do? Put a bent glass tube. Put a bent glass tube. But in this glass tube, now that we have fastened this region, okay, now in this glass tube, what is important? First of all, you have to use a manometer. What is a manometer? A scale, a graduated scale. So I'm saying that this is a manometer, which is giving me some readings, which is giving me some readings, 0 and then 5 and then 10 something. It is giving me some readings here. So now that we are talking about this manometer, what is a manometer? A graduated scale. Next, what are we doing? Next, we are filling an amount of mercury here. We fill certain amount of mercury here. We fill certain amount of mercury. What are the reason of filling mercury? Because mercury does not stick the wall. It is transparent. Will it move? Will it mix with water? Never. So after a particular period of time, we had set this arrangement, the same that I show you there. So we have made this arrangement. After making this arrangement, we wait for a particular period of time. In some couple of hours, what are we going to observe? That there will be some clear liquid getting filled up in this tube. And this will gradually happen, filling up, filling up, filling up, filling up, filling up in this tube. And it will take a particular period of time. It's a slow process. In some couple of hours, in some 4 to 6 hours, if we are talking about a well-watered plant, yes, the entire tube gets filled with the transparent liquid. What is this transparent liquid? Actually, it is sap. It is the sap that we are talking about. Then what will happen to this mercury now? It will be pushed. It will be pushed, no? So this level of the mercury will be pushed now, and this level of the mercury starts going up. This level of the mercury now starts going up because this is pushing the level of the mercury. So now that we are talking about that this liquid which has started pushing the level of the mercury, and the mercury is showing a particular mark. Initially, it must have reached the 0. And then we wait it for another 1 hour, and then we got to see that now it has reached to the 5 mark. Wait for another maybe 1 hour, and it reaches to the 10 mark. So what is happening? The rise in the level of the mercury indicates the amount of the sap released from the stem. Whatever is the amount of the sap released from the stem, that is only going to push the mercury, and the mercury level goes up. Now we are trying to understand that yes, first thing is very clear that the moment you talk about cutting the stem, there is a particular clear liquid which is rising up or releasing from the stem, oozing out from the stem. So there is a particular clear liquid oozing out from the stem, and now which is getting filled up in the glass tube. So what is this clear liquid? Obviously the cell sap. Oh, water kahan se aa raha hai? From where is this cell sap coming? Because the roots are constantly absorbing the water from the soil, and this water is becoming a component of the plant cells, and that is now we are talking about the cell sap. What is cell sap? Water plus minerals. That's the sap. So water plus minerals. Now we are trying to understand that what is this pressure? What is this pressure? The pressure which is responsible for moving the liquid and moving the level of the molecule. What pressure are we talking about? There is a pressure which is getting exerted at this level of the root cell. What is this pressure? We talk about root pressure. Let us try to define this pressure. It is only a particular type of pressure which is exerting here or happening here, which is responsible for the oozing out of this liquid and pushing the level of the molecule. So what is this pressure? Root pressure. Let us define the pressure exerted. The pressure exerted at the root cells due to the continuous inward movement of water, continuous inward movement of water and cell-to-cell osmosis, which helps in the ascent of sun. Now we have to understand this. I try to understand definition of root pressure. ये लिखना है. This you will have to understand. समझ के याद करो. What is root pressure? The pressure which is exerting at the root cells, which is allowing inward movement of water. You already know what process is going on in the root cells. Endosmosis is happening. So here two things are happening. First, I will say that why did I write inward movement of water? Because every time the water is entering the root cells, a process known as endosmosis. And this water, by the way of the root, is entering into the stem. This water, by the way of the root, is entering into the stem. Every root absorbing water by endosmosis, and this is the water which is diverted upwards. This is the water which is diverted upwards. This is the water which is diverted upwards. So if I have to talk about a plant, the continuous movement of the water upwards, what do we call that process as? Ascent of sap. That is what is called ascent of sap. What is the meaning of this? Ascent of sap. Kisi bolenge? What is ascent of sap? The upward movement of water or the upward movement of sap. So if I now talk about a plant and then I understand that this plant has got all the branching patterns. So it is very clear that once I talk about this entire plant, the upward movement which is happening by what type of tissues? The upward movement of the water with the help of the vascular bundle known as xylem. So yes, if you have to start from the roots also have got the xylem tissues. So this entire spread out system of the roots, they will also have the xylem tissues. So first of all, this, then from the root to the stem, then from the stem to the branches, and then to the leaves. So that is what we are talking about. This is what is ascent of sap. What is the meaning of this ascent of sap? Upward migration of water and minerals by the xylem. So here, root pressure is exerting. The entire pressure is responsible. When we talk about what is the pressure which is responsible for the ascent of sap, your answer should be root pressure. Which pressure is responsible for the movement of the water in the upper parts of the plant? That should be always root pressure. There is one more definition. Let us go to that first. There is one more definition of the root pressure. Here I am showing you again that if you are trying to understand this figure, you get this figure in your paper. And whatever I told you right now, the water entering into the root hair cells, then by a series of cells, they will enter into the stem. The water from the soil by a series of cells will enter into the stem and then fall into the xylem cells. What are those series of cells? Let us learn right here. The water will enter from the soil to the epidermal cells, the root hair cells. From the root hair cell, where they will go? To the cortex. Large number of cells arranged in a series. Cortex. These cortex cells, try to understand, the moment I talk about the water which is moving from the epidermal cell to the cortex cell, a single cortex cell, what will be the behavior of this cortex cell? Initially, it is becoming turgid, receiving water. Then what will happen? Once it is turgid, ये अपना excess water दूसरे cell को दे देगा. So this turgid cell has become flaccid. So since it has become flaccid, that's the reason it will again take water from the epidermal cell. So that's the reason when we say ये जो root pressure है कैसे? How does it work? Root pressure also works by the alternate turgidity and flaccidity of the cortex cells. Always to remember this. You can give reason questions. Root pressure works with the alternate turgidity and flaccidity. Is that true? Yes, it is. क्या लिखना है? Just now I said when the water is entering this cell, अब जब ये cell turgid हो गया है, the water will move into the cortex cell. अब ये cell भी turgid हो गया, it will give the water to the next cell. Now that it has given water to the next cell, ये अभी flaccid हो गया ना? Once it has given water to the next cell, now the turgid cell, the cortex cell has become flaccid. अब ये जो flaccid हो गया है, now it will again draw water. So the same is the story for this, same is the story for this, same is the story for this. In this entire sequence, एक cell, cortex cell, एक बार turgid होगा, next moment में flaccid होगा. एक बार फिर से turgid होगा, next moment में flaccid होगा. So every cortex cell undergoes alternate turgidity and flaccidity, and that is how the water is moving forward. इसमें भी which pressure is responsible? Root pressure. Again, see the definition. What is it? Pressure which is developed at the root cells, which is allowing the water to enter by osmosis and then allowing the further movement. So ये further movement कैसे होता है? How is the further movement? Cell to cell osmosis. एक बार turgid, दूसरे बार flaccid. हर एक cell is suffering that. Then what? From the cortex, where will the water go? Take that diagram in your book. Cortex say that water will go to the endodermis, and then it will enter the xylem cells, and then it will enter the xylem cell that I showed you here. Take that diagram in your book. You are to remember the sequence of cells: epidermal cells, cortex cell, endodermis, and then the xylem. This is the manometer experiment. Manometer experiment for the root pressure is there in your book. Manometer. This is known as the manometer experiment for the root pressure. Okay. Read till here. What is the question for your next exam? School.
Ma'am, it is transpiration, absorption, exclusively endocrine and population.
Achha, okay. Maybe tomorrow is a test or?